Now, we'll be welcoming a leading researcher in advanced imaging technology. Now, please welcome Professor ZHANG, Research Professor in the Department of Biomedical Engineering at Zhejiang University. Professor ZHANG will be talking about advancing chemical exchange, saturation transfer imaging, bridging scientific discovery, and global clinical translation.
Thank you for the introduction, and thanks to Dr. Kenny Chan for the kind invitation. This is the title that Kenny gave me. I'll try to stick to it very loosely, I could say that. I think I don't quite fit in this session. This is more industry session. I'll try to do my presentation slightly differently. This was my original outline of the talk. I will start on a brief background of the CEST imaging, the principles of CEST imaging. I will go into technical details, but I think I'm going to skip this part because it's too much technical. Then I will go through some clinical values of CEST imaging. I think I will try to focus on this part because for the industry people, this might be more interesting to you guys. And then I will wrap up with a brief summary. Let me start with the principal part. So MRI can really provide a very rich variety of image contrast. So with the same hardware, with the same scanner, when we run different sequences, when we run different acquisition techniques, we can get these many beautiful MRI images. For example, these T1 weighted and T2 weighted images that we use in daily clinical practice. And also we have some more advanced techniques like diffusion MRI or functional MRI. You can study the brain structures, microstructures, and the brain functions. And the technique I'm going to talk about is called CEST, chemical exchange saturation transfer imaging. So CEST MRI is slightly different from the other images that you see here. So for the regular MRI scans, we detect the signals from the water molecule in the human body. For a regular, proper fit person, the water molecule make up 70 to 80% of our body weight. That's what the regular MRI techniques detect. That's where the image comes from. But the CEST MRI, it can detect the non-water molecules, like the proteins or metabolites in our human body. So that's the unique part or unique advantage of CEST MRI. Of course, with the spectroscopy, the technique that Dr. Peter Barker talked about yesterday, it can also detect metabolites in the human body. But it's usually a non-imaging technique. It can only acquire data from one voxel or tens of voxels. It's not really, it's not really an imaging technique in a clinical routines. So that's in the CEST MRI, because it can detect these non-water molecules, we usually call it molecular MRI in the field. So here I will briefly talk about the principles of the CEST imaging. So what you see here are two peaks. One is a small peak that stands for the metabolite signal. The other is a bigger peak that stands for the water signal. This is really just a schematic figure. In reality, the water signal is over 1,000 times stronger than this metabolite signal. So you can't really see the small peak because it's buried under this huge water signal. But there's a natural phenomenon. So the hydrogen atoms or the hydrogen protons that we usually call it in MRI, the hydrogen atoms exchange between these two pools, these two molecules. One is a metabolite molecule, the other is a water molecule. It's really just a natural phenomenon. They exchange hydrogen atoms. So based on this natural phenomenon, we can devise a new experiment. We can acquire two sets of MRI images. One is a regular, like the structural MRI, like a T2 weighted MRI that we obtain in the routine clinics. That's the first set MRI image. Then the second set MRI image, before we start the second set of acquisition, we can crush this metabolite signal. We can saturate the metabolite signal. And this crushing effect will be transferred to the water signal because of the exchange of hydrogen atoms between these two pools. Then after a while, we will see a decrease in the water signal. Then we can acquire the second set of T2 weighted image. Then the subtraction between these two T2 weighted image will give us information about the the metabolite signal. I mean, this is how we do the CEST imaging acquisition. So basically, this is the subtraction method, or we detect this metabolite signal indirectly. We actually are detecting the change on a water signal. So we are detecting this metabolite signal indirectly because this is a signal amplification process. So as you can imagine, as you can see, the exchange protons, hydrogen protons between each other. There's an exchange rate. So say it's 100 hertz. That means these two molecules, they exchange hydrogen 100 times per second. So another way you can imagine is it amplifies the low signal metabolite signal 100 times per second. So this is a signal amplification mechanism that's quite special. It can amplify the low concentration signal by tens to hundreds of times. So that's why we can do high resolution imaging. And the signal difference, that we detect, or the subtraction between these two T2-weighted image, will tell us information about the concentration of the metabolite pool, and also the exchange rate between these two pools. And in an in vivo environment, the exchange rate depends on pH. So with the CEST-MR technique, we can detect both the concentration of the metabolite or the proteins, and also the pH in the in vivo environment. So with this basic principle, I will skip the technical part directly to the potential applications. I think they might be more interesting to the industry people. So what we can do with the CEST-MRI, the reason we work on CEST-MRI is that we think it provides extra clinical values beyond structural MRI. So the first example that I will give you is about glioma grading. So what you see here is a brain tumor patient. The images, these three, like a grayscale or black and white images are from the routine MR scans. What you see here, this is a T2-weighted MR image, this is a FLAIR image, and this is a post-contrast T1-weighted image. And usually, like when the clinicians see this post-contrast T1-weighted image, they don't see a enhancement in the tumor region. So there's no signal increasement after the contrast injection. The radiologist would tend to think this is a low-grade glioma. But in fact, this is a high-grade glioma. This is a WHO grade 3 glioma. Because, I mean, that's based on pathology. But based on CEST-MR image, what you see here in the tumor region, sorry, it's very difficult to use. In the tumor region, you see a signal increase that's related to protein. That's a protein CEST-MR image. So it's related to the overexpression of protein signals in the tumor. So that's an endogenous signal. So it doesn't depend on the external contrast agents. So with this CEST-MR technique, it can help diagnose the glioma grades. I think this is a very early on successful application that's coming out from Dr. Jing Andrew at Johns Hopkins University. The second example is still about the glioma, but this is about the treatment effect. For these glioma patients, especially the high-grade glioma patients, they usually undergo surgical resection and also chemoradiotherapy. But unfortunately, the glioma will recur at some point. So it's a clinical question to diagnose whether it's a tumor recurrence. And the real challenge is to separate the true tumor progression or the tumor recurrence from like the treatment effect or pseudo-progression. So these are cases, these are two representative cases. So the first row is a true tumor progression or the tumor recurrence. The second row is a pseudo-progression. So it's due to treatment effect. It's not a real tumor. But on the conventional structural MR images, this is the first column is the post-contrast T1-weighted image. You see very strong enhancement. I mean, this enhancement is what usually the radiologists they look for. When you see this high enhancement, it usually means something bad. And also it has a very big like a tumor, and also edema region surrounding the tumor. But you can't really tell from the structure, conventional structure MRI, whether this is a true tumor or not. But on the protein CEST MRI, for the tumor recurrent case, you see clearly high signal in the tumor region, and that represents the active metabolic activity and overexpression of proteins. In the pseudopercussion case or the treatment effect case, you see a low signal in the tumor region because it's not a real tumor. I mean, this is also a kind of like a successful application for diagnosing the treatment effects. Also coming out from Hopkins. This is the last example related to glioma. I'm going to show you. Still coming out from Hopkins. So we can use CESS MRI to guide biopsy. So this is a newly diagnosed glioma patient. We see this T2 flare or T1 weighted image and also post-contrast T1 weighted image. There's really no enhancement in the tumor. And it's a fairly uniform tumor. Then a real challenge, a practical challenge is where to biopsy the tumor. You can't take out the whole tumor during the biopsy stage. But if you look at the CESS MRI, inside the tumor, it's clearly quite inhomogeneous. It has a high protein signal, CESS signal region, and a low protein CESS signal regions. And it guided a biopsy. So they took one biopsy from the high CESS signal region. That turned out to be WHO grade 3. They also took out one biopsy from the low CESS signal region. It turned out to be WHO grade 2. I mean, of course, this patient was diagnosed as high-grade glioma in the end, but it tells us there can be multiple glioma grades within a single lesion. I think it's also a quite promising application. Certainly, we can go beyond tumors. This is a study done in my group where we are trying to use CESS MRI to detect epileptic lesions. So the image that you see here are from patients with epilepsy secondary to tuberous sclerosis. So the patients have tumors inside their brain. And these tumors show up as hyperintensity or high signals in a regular structural MRI. As you can see in this red circle region and also in this black box region. That's like the tumors that we can detect with this structural MRI. But I want to draw your attention to this yellow arrow region. The yellow arrow region looks completely normal on structural MRI. So basically with the structural MRI, there's nothing wrong. There's no tumor, there's no lesion in that region. But with the CESS MRI, you can definitely detect something abnormal, some abnormal signal elevation in this yellow arrow region. And that's, I think that means we can detect something that you don't really see on FLAIR images. We can detect some extra lesions. I think, and also through like post-processing, we think this is due to glutamate signal that's very, very much related to epilepsy. We can also use CESS MRI to study strokes. Yesterday, Dr. Peter Barker talked about using spectroscopy to study strokes. We can also, in stroke, the stroke lesion produces lactate, and that lowers pH. We can detect this pH change with the CESS MRI. As I mentioned in the principal part, CESS MRI can detect concentration changes and also pH changes. So this is a patient that we scanned in our affiliate hospital. This is a wake-up stroke case. So the patient didn't go through a thrombolysis treatment. That's why we could scan the patient with MRI. On day one, we scanned the patient with a diffusion MRI and also T2-weighted structural MRI. We can clearly see this lesion on diffusion-weighted image, but you don't really see that on the T2-weighted image, or at least it's extremely hard to see that unless you are extremely experienced. But we clearly see a pH decrease or pH-related lesion in the CESS image or pH-related to CESS MRI image. Then on the day two, we followed up the same patient. We see a increased lesion size, and also now we start to see this lesion on the T2 and the T2-weighted MRI. Of course, on CESS MRI, we see a further decrease in the pH-related signal and also an increase in the lesion size. I think from this result, I want to show you that CESS MRI can detect these stroke lesions earlier than T2-weighted MRI. We have, in our patient cohort, we haven't seen the CESS MRI could detect lesion earlier than diffusion MRI. But in literature, in the rat models, in some cases, CESS MRI can even detect lesions, stroke lesions earlier than diffusion MRI. That's quite actually promising. We can use CESS MRI not only to detect diseases, we can also detect brain functions on healthy volunteers. This is our recent study. We're using CEST MRI to study brain functions. So in the MRI field, there's a very famous technique called both fMRI or blood oxygen level dependent functional MRI. So to use both fMRI to study the brain activations. So the maps that you see here are the activation maps. It shows which brain regions get activated. So this volunteer goes through the visual stimulation. So basically, we play a flickering checkerboard image for the volunteer. So on the both fMRI, it shows the activation regions in the visual cortex, quite extended visual cortex. But now we are using glutamate-weighted system MRI to study the same visually stimulate brain regions. We see a much smaller activated region. It's more concentrated within the primary visual cortex of V1. The both fMRI is very much related to blood flow. As we all know, blood flow comes from the heart, so it's not really localized. It's not endogenous in the brain. But with glutamate-weighted system MRI, the glutamate signal is really in the brain, right? It's not coming out from the heart. So in this case, we are showing that the CEST fMRI is more spatially specific than the conventional both fMRI. We are also trying to extend this technique to study more neuroscience and mental health diseases with the CEST fMRI. And lastly, this is a fun slide. We can even apply CEST fMRI to plant sciences. We can go beyond like in vivo situations. So this is a study coming out from Germany. So what you are seeing here are pea seeds. So these are pea seeds. As we all know, pea seeds, they got a lot of sugar and amino acids. These things can be detected by CEST fMRI. So you see here, these are very nice high-resolution CEST maps for the sugar and for the amino acids. Of course, because this is a pea seeds, you can also use spectroscopy with a very long scanning time to get the similar sugar and amino acid maps. But clearly, they have much lower spatial resolution compared to the CEST maps. And also, the authors reported a 200-fold signal increasement with the CEST technique compared to the spectroscopic technique. So again, I mean, this is consistent with the unique advantage of CEST MRI. It can amplify the signal by tens to hundreds of times. Okay, I think I would just really skip my technical part to wrap up my presentation. I think I'm just trying to show that CEST is a novel molecular MR technique. It can detect the non-water signals, like proteins and metabolites, without the injection of contrast agents. I skipped the technical part, but in my group, we work on the acquisition, reconstruction, and analysis methods related to CEST fMRI. We have developed techniques for stabilized and accelerated CEST scanning. Also, we invented a method like KP CEST vegan for reconstructing images from unsampled data. And also, we developed an analysis method to extract these lesion regions more clearly, more robustly than the conventional methods. So to sum up, I'm trying to convince you, I hope I have convinced some of you that CEST fMRI shows great promise for imaging patients and discovering novel contrast without contrast agents. It has a unique added value compared to conventional structural MRI. With that, I'd like to thank you for your attention.
Professor ZHANG, ladies and gentlemen, please remain on stage. Now, Professor Chen would like to present to you a souvenir. Right, some photos first.